Let's talk today about the world of genetics, and specifically DNA, and how it became known as the genetic material. Now, for a long time, no one had any idea exactly what this genetic material was. Now, eventually, by the early 20th century, it had been theorized to be chromosomes, as some sort of model of inheritance of traits. But the problem from that is that chromosomes are complexes, and you have proteins, and you have DNA. Plus, once you look at cells, you have all sorts of giant macromolecules, so you can't really tell what's doing what. And when you come to the chromosomes, well, you don't know if it's the proteins or the DNA that's doing all the work. So, these are some experiments that determined that DNA is the genetic material. Let's go to 1928, and you have a scientist by the name of Griffith. Now, he takes Streptococcus pneumoniae, and it has two different strains to it. You have the S strain, which is a smooth strain. It has a hard outer shell. Well, it has a coated shell. That means that it's virulent. And you have the R strain. This has a rough outside. It doesn't have that coated shell, so it isn't virulent. It can be killed when it goes into the system. So Griffith takes these S strain bacteria, injects it into mice, and, well, he finds they die. He gets the R strain, injects it. Well, he found out this time that the mouse lived. So he kills the S. He says, let's see what happens when I kill this S strain bacteria. Injects it. And this time he found the mice lived. So he says, why don't I take this heat-killed S, combine it with some R, inject it into some mice, and see what happens. And this time, well, he found out the mice died. So now he's saying there's something inside this bacteria that he calls a transforming principle. And that's what he concluded from his experiments, that there is some sort of transforming principle. He didn't say what it was. I'm sure he had his own ideas, but there is some sort of transforming principle inside the S that allows it to make R strain into S strain and kill the mice. So let's jump all the way to 1944. And you have Avery, McLeod, and McCarty. So they want to know what part of the cells is the part that is this transforming principle that Griffith found. So they take the same bacterial cells that he had, they lyse them, and they separate them out. So they have a test tube now filled with all sorts of cellular components. You have lipids, you have proteins, you have RNA, and you have DNA. They mix this with some living R. They put it in a little petri dish, and, well, they find that they get living S colonies. Now, by this time, by the 1940s, they discovered all sorts of enzymes that can selectively destroy different macromolecules found in cells. So they take this cellular extract and they treat it. They treat it with a lipase, mix in with the R, and they still found S colonies. So that's a yes. So now they take the proteins, treat it with the protease, and, well, they still found S colonies. And you take the RNA, destroy that with an RNase, and they still found S colonies. So then they destroyed the DNA with the DNase, and this time they found no S colonies. So Avery, McLeod, and McCarty concluded that DNA is that transforming principle that Griffith talked about. But because this is the world of science, naturally we're skeptical, so other people are like, well, maybe there's something wrong with this experiment. This isn't conclusive enough to say that DNA is in fact that transforming principle. Maybe other stuff was mixed in, maybe there's something wrong with the setup, and you weren't able to selectively control exactly which macromolecules were destroyed. So let's jump ahead 8 years now. It's 1952, and you have Hershey and Chase. And they are looking at a virus, T2 bacteriophage. And this is a virus that is made of DNA and proteins. And they take this and they say one of these things is what causes this bacteriophage, this virus, and what it does is it hijacks bacteria and it causes the bacteria to make new virus cells. So they're saying one of these two things is responsible for making the bacteria make new virus cells. So they take virus cells and they have one treated with radioactive phosphate, and they have those labeled virus cells. And they have another one with the sulfur. Now, the phosphate will make its way into the DNA because of DNA's sugar phosphate backbone, while the sulfur will make its way into the proteins. So what they're saying is, when they have these viruses and they mix the virus and the bacteria together, and then they look at the outcome, that the bacteria will be labeled with either this radioactive phosphate or this radioactive sulfur. So they put these in test tubes, they inject bacteria into the test tubes, let them mix together and have successive generations, and they centrifuge them out, and what they find is down here in the pellet that has bacteria, well, they find with the phosphate that the bacteria is labeled with the phosphate. But in this one, they have the bacteria pellet. The bacteria isn't labeled with the sulfur. The sulfur was still floating around in the supernatant. So therefore, from this experiment, Hershey and Chase were able to conclude that DNA is this genetic material, because it is what made its way into the bacteria and is what transformed it into making new virus cells. And after this, well, the world of genetics, with the conclusive evidence that DNA is a genetic material, exploded into finding information about traits and about heritability and about everything about how DNA works. So this was really groundbreaking work to the world of genetics, discovering that DNA is the genetic material, is that transforming principle by which traits are handed down from parents to offspring.